Welcome to Bookfulness. This is our deep dive on episode 16. We just finished unit 3, really digging into, the processes of evolution, how natural selection works within population.
Right, the micro level stuff.
Exactly. Today, we kick off unit 4, which is macroevolution and the history of life. Big picture time.
Yeah.
And we're starting with something really foundational, how scientists actually build the evolutionary family tree.
The phylogeny.
That's it. If microevolution is like looking under the hood of the engine, macroevolution is, well, it's the whole road trip, the history of life's journey.
Nice analogy.
So our mission today is to get through chapter 16, Phylogeny, the unity and diversity of life. That's pages 401 to 427 in the textbook.
Okay.
We're going to learn how to read these maps, these historical diagrams of life, and importantly, tackle some of the really deep technical challenges in making them.
Right, because it's not always straightforward.
Not at all. But also, we'll see the incredible things, sometimes really surprising things we can figure out once we do have a reliable phylogeny. Okay, the sources are here. We're ready. Let's unpack it. We're shifting gears now, moving from those small generation-by-generation changes inside a species to the huge historical patterns above the species level. And yeah, the first tool you absolutely need for that is this map of relationships, the phylogeny. All right, let's start with the basics then. What exactly is a phylogeny? What are we looking at?
So the core definition, it's pretty precise. A phylogeny is the history of descent.
Okay.
Of a group of taxa.
Yeah.
Now, taxa could be species or maybe whole genera, families, orders, whatever level you're interested in.
Right.
It shows their descent from their common ancestors, and it's usually shown as a branching diagram, like a tree, which is fundamentally a hypothesis.
A hypothesis about history.
Exactly. It shows the order of branching, who split off from who and when, and sometimes, if you can calibrate it, it can even show the absolute times of divergence. Millions of years ago, perhaps.
And this map, this hypothesis, it's the foundation for some really profound statements in biology, right? The source mentions these outrageous claims that just sound, well, wrong on the surface.
Oh, absolutely. Think about the idea that chickens and crocodiles are close relatives.
Yeah, that one always gets people.
Right. To anyone just looking, they seem completely different. One flies, one crawls, scales versus feathers.
Totally different animals.
But the phylogeny forces you to accept it. Their most recent common ancestor is much more recent than, say, the common ancestor crocodiles share with lizards. Or, the now completely established fact that birds are dinosaurs, not just descended from, but are a type of dinosaur.
So the phylogeny isn't just suggesting these things. It's providing the actual historical evidence, the framework to test and confirm them. It traces the lineage back, shows the sequence of events.
Precisely. Without that historical map, those connections seem, well, maybe just guesswork, speculation. But with the phylogeny, they become testable scientific conclusions. But, okay, how do we build the map? That's the real question. We can't just go by looks, by superficial resemblance.
No, definitely not. We need objective evidence for how related things are. And the mechanism for that, it relies completely on identifying what the sources call synapomorphies.
Sure.
Shared derived characters.
That's the key term, synapomorphy. It's a character state that means a trait could be physical, could be molecular, like DNA.
Okay.
That is derived. Derived means it evolved within the specific group, the lineage we're studying.
So it's new for that group.
Right. And critically, it has to be shared by 2 or more taxa in that lineage because they inherited it from their common ancestor where it first appeared.
Okay, so let me see if I get this. If having 5 fingers is the ancestral state for all land vertebrates, tetrapods, and 2 species both have 5 fingers. That doesn't really tell us they're super close relatives within that bigger group.
Exactly. That's just an old ancestral character. A plesiomorphy is the technical term. It doesn't help resolve relationships within the group.
Got it.
It's the new shared evolutionary innovation that matters for figuring out the branching pattern. And these shared derived characters, these synapomorphies, they are what define a clade.
Clade.
And a clade is like the gold standard in modern classification. It's what we call a monophyletic group. That means it consists of an ancestor and all of its descendants. One complete natural branch of the tree, no exceptions.
The source uses the classic example of mammals, which I think makes this really clear.
Yeah, it's a great one. If you take a group of mammals, say a human, a rat, a kangaroo, they're all united by certain traits, hair, obviously, mammary glands, and a really specific one, having only a single bone in the lower jaw, the dentary bone. Now, to be sure these are derived characters for mammals, we need to compare them to outgroups. These are taxa that are related, but definitely outside the group we're focusing on, like a crocodile or a bird or maybe a salamander.
And those outgroups, they don't have these features. Crocodiles have scales, not hair, lay eggs, and they definitely have multiple bones in their lower jaw.
Right, so the absence of hair, mammary glands, and that single jawbone in the outgroups confirms that these traits are evolutionary innovations. They popped up in the common ancestor of mammals.
Ah.
So they are synapomorphies that define the mammalian clade. They tell us who belongs together in that branch.
Okay, that makes sense. But in the real world, especially now with huge DNA data sets, you must end up with tons of characters, right? Hundreds, thousands maybe? And the data probably doesn't always line up perfectly. You get conflicting signals.
Oh, absolutely. It's rarely clean.
So how do scientists choose the best hypothesis, the best tree, when there are multiple possibilities, maybe even conflicting ones?
Right. That brings us to the most fundamental principle used, historically at least, for estimating phylogenies, the rule of parsimony.
Parsimony. Okay. Sounds like being frugal.
Kind of. It's based on, well, computational simplicity, but also a kind of biological likelihood. The rule simply states, choose the tree that requires the fewest evolutionary changes, the minimum number of steps like mutations or trait changes to explain the character states you actually observe in your species.
So basically, we're penalizing hypotheses that require a trait to evolve independently multiple times, or to appear, then disappear, then reappear again.
Exactly. You want the simplest historical explanation, and the textbook has a great illustration of this using the bipedalism problem. That's figure 16.2.
Bipedalism, walking on two legs.
Right. If you looked only at that one trait, walking on two legs, you might be tempted to group humans and birds together, because both do it.
Yeah, you might. But we know, or we strongly suspect, that bipedalism evolved independently in birds and in our human lineage. It's a misleading similarity, what we call homoplasy. We'll get more into that later.
We will. But let's just play the parsimony game for a second. Imagine we did group birds and humans as closest relatives based only on bipedalism. What would that tree imply? The computational cost, in terms of evolutionary steps, becomes enormous. We'd have to assume their immediate common ancestor was, let's say, somehow mammalian-like to link to humans, but then the bird lineage would have had to lose a whole suite of complex derived mammalian traits, things like hair, mammary glands, the single jawbone we just talked about.
Oh, okay.
Reversals.
Yeah, 4 crucial evolutionary reversals shown as blue crossbars in the figure. They'd have to lose those key mammalian traits just to end up looking like a bird.
Yeah.
That's 4 extra complex steps.
That seems unlikely. Very costly in evolutionary stuff.
Highly unlikely. The correct tree, the more parsimonious one, shown in figure 16.2a, groups humans with other mammals. This tree only requires 2 independent origins of bipedalism, one on the bird branch, one on the human branch. That's it.
So 2 steps versus, what was it? The original 2 plus 4 reversals, 6 steps. Exactly. 2 steps is much simpler, much more parsimonious than 6 steps involving complex losses. Parsimony is like our evolutionary accountant. It rejects the hypothesis that requires the most sort of unlikely transactions, especially losing complex traits that took a long time to build up.
That makes sense.
Okay.
It's not just about the number of steps, but the biological plausibility. Simplicity often reflects reality, but you mentioned it struggles when evolution isn't simple.
I did, and that's the perfect segue into the challenges. Parsimony is a powerful rule of thumb, but it has its limits.
Okay.
But before we dive into those challenges, just a quick reminder for everyone listening, all the figures we're talking about today, figure 16.2, and the ones coming up, they're all linked in the description file, so you can pull them up and follow along visually.
Good point. Definitely helpful to see these trees. Okay, let's face it then. Evolution isn't always neat and tidy. It's messy. What's the biggest challenge that makes simple parsimony sometimes? Well, fail.
Without a doubt, the biggest headache is homoplasy.
Homoplasy.
Yeah. If parsimony is our ideal logical accountant, homoplasy is like, well, it's like evolutionary coincidence screwing up the books. Homoplasy is the independent evolution of similar traits in different lineages. The key thing is, the similarity is not because they inherited it from a recent common ancestor. It's not homology.
So evolution repeating itself, basically. Finding the same solution multiple times independently.
Exactly. Nature hits on the same idea more than once. The source breaks homoplasy down into a few types, like convergent evolution, parallel evolution, and evolutionary reversal. Let's focus on convergence and reversal as their super common sources of confusion.
Okay, convergence first.
Convergent evolution is probably the easiest to grasp. It's when distantly related lineages independently evolve a similar shape or function, usually because they face similar environmental pressures. The classic example is fins.
Fins.
Yeah, think about the fins of a shark, a dolphin, and maybe an ancient marine reptile like an ichthyosaur. They all have that similar streamlined shape for moving through water.
Right. But sharks are fish, dolphins are mammals, ichthyosaurs were reptiles.
Exactly. Very distant relatives. They didn't inherit that fin shape from a recent common fish-mammal-reptile ancestor. They each evolved it independently because it's a good solution for swimming fast.
The plant example in the source, figure 18.1, that's also really striking. Desert plants looking identical on different continents.
Oh, yeah, the cacti and euphorbias. It's amazing. Cacti are only found in the Americas, the New World. And certain species of euphorbia, they're found in the Old World, Africa and Asia. But they live in really similar arid desert environments.
And they look the same.
Almost identical. Both groups independently evolved to be leafless, succulent plants. They have thick, fleshy stems that store water and do photosynthesis. It's all about minimizing water loss in the desert.
Wow.
But if you trace their ancestry using other features, or especially DNA, you find they belong to completely different, very distant plant families. If you just group them by their succulent look, you'd be creating a totally artificial, polyphyletic group based purely on convergence. Homoplasy tricked you.
Okay, so that's convergence. What about evolutionary reversal, where a group goes back to looking like an ancestor?
Right. The example in figure 16.3 is wing loss in insects. This is where a lineage loses a derived trait and reverts to an ancestral state.
How does that work with insects?
Well, if you look at the insect phylogeny, the really early basal insects like silverfish or bristletails, they were wingless. That's the ancestral condition for insects.
Okay.
Then later, wings evolved in a common ancestor of most insects. Flight was a huge innovation.
Yeah, obviously.
But then, in many different separate lineages, think about fleas or lice or even worker ants, wings were lost again. Independently. They reverted back to the ancestral wingless state.
So winglessness in fleas and lice isn't because they're super closely related.
Exactly. If a scientist only looked at that one trait, being wingless, they might lump fleas and lice together.
But that would be wrong.
Totally wrong. Again, homoplasy fooling us. We know from lots of other evidence, especially DNA, that lice are actually related to true bugs, the hemiptera. And fleas are related to flies, the diptera. The wing loss happened completely separately in those two lineages, and in many others too. It shows why relying on just one or two traits is dangerous. You need lots of characters, ideally molecular data, to cut through the noise.
Speaking of DNA, molecular data sounds like it should solve everything, right? Millions of characters. But it brings its own set of problems, its own technical nightmares. The source mentions saturation. What's that about?
Saturation. Yeah, that's a big issue when you're looking at really deep evolutionary time, like hundreds of millions of years.
Okay.
Imagine one specific site, one letter, in a long DNA sequence. Let's say it starts as an A. Over vast stretches of time, it might mutate to a C, then later maybe it mutates again to a T, and maybe just by chance, it mutates back to an A again later on.
So it can change multiple times at the very same spot.
Exactly. When this happens repeatedly at the same site across different lineages, the number of observed differences between two really ancient species kind of hits a ceiling. It saturates. You can't accumulate any more observable differences because new mutations are just writing over previous changes.
It's like painting over old graffiti.
Yeah.
You eventually just see the top layer, not all the history underneath.
That's a great way to put it. Old divergences can end up looking deceptively recent because the sequence is so crammed full of changes that the signal of true ancient difference gets lost. It gets overwritten.
So how do we deal with that?
Well, one way is to rely on the fact that different parts of the genome evolve at very different speeds. The source calls this differential rates, and it's shown in figure 16.5.
Right, so we use the fast-ticking parts of the clock for recent history and the slow-ticking parts for deep history.
Pretty much. For recent divergences, say within the last 75 million years or so, we look at DNA sequences that evolve really quickly. A classic example is the third position in codons of protein coding genes.
Why the third position?
Because mutations there often don't actually change the amino acid that the codon codes for. They're called synonymous mutations. Since they don't change the protein, they're often effectively neutral, not strongly selected against, so they can accumulate quite rapidly. Good for telling apart closely related species.
Okay. And for the really deep branches, the ancient splits.
For deep history, we need the opposite. We rely on the slowest evolving sites. like the second codon positions.
And why are they slow?
Because a mutation at the second position almost always changes the amino acid. And changing an important protein is often bad for the organism, so those mutations tend to be eliminated by natural selection, what we call purifying selection. These sites are highly constrained, they change very slowly, preserving the ancient historical signal over hundreds of millions of years. Allows us to resolve those really deep branches in the tree of life.
That's clever.
Yeah.
Using different data types for different timescales.
Mm-hmm.
But even if we pick the right clock speed, we can still run into problems where different genes, even within the same organism, seem to tell conflicting stories about history. Now, we're not just talking about misleading traits, but actual chaos in the genetic record itself.
Yes, this is where modern phylogenetics gets really, really complex and honestly quite fascinating. There are two main reasons why gene histories might conflict with each other, or with the true species history. The big ones are incomplete lineage sorting, ILS, and introgression, which is basically hybridization or gene flow between species.
Okay, let's take incomplete lineage sorting first. ILS, what's going on there?
ILS happens when a species already has multiple versions, multiple alleles of a particular gene floating around in its population. That's called a polymorphism.
Right, genetic variation.
Exactly. Now, if that species splits into two daughter species relatively quickly, that ancestral polymorphism, those different alleles, might persist through the speciation event. Both new species might inherit multiple old versions of the gene. Then, later on, just by chance, one daughter species might lose one allele, and the other daughter species might lose a different allele. When that happens, the history of that specific gene, the gene tree, might not actually match the history of the species splitting the species tree.
Can you give an example?
Sure. Let's say species A and species B are truly the closest relatives. They split most recently. But imagine that before they split, their common ancestor had two alleles, let's call them red and blue, and maybe species C, a more distant relative, only had the blue allele.
Okay.
Now, A and B split. Maybe species A happens to keep both red and blue for a while, but eventually loses the red one, keeping only blue. And species B keeps red and blue, but eventually loses the blue one, keeping only red.
So A ends up with blue, B ends up with red.
Right. But remember, the more distant species C also has the blue allele. So if you build a tree based only on this one gene, it looks like species A and species C are closest relatives because they both share the blue allele. But the true species history is that A and B are closest. The gene tree disagrees with the species tree.
I see. The gene history got sorted differently from the species history because the split was fast and there was ancestral variation.
Exactly. ILS is super common in groups that diversify really rapidly. The classic example is the cichlid fishes in the big African rift lakes like Lake Victoria or Lake Malawi. They speciated incredibly fast into hundreds of species.
Yeah, amazing radiation.
And because it happened so fast, many of their genes didn't have time to sort out cleanly into groups matching the new species. So you get tons of conflicting gene trees. It makes figuring out their true relationships a real challenge.
Okay, that's ILS. What about the second reason for conflict? Introgression, or hybridization. Gene flow between species.
Right. This is when genes actually move from one distinct species into another after they've already diverged, usually through occasional hybridization and backcrossing. This actively mixes genetic material and really blurs the historical lines.
And the source has this incredible, really detailed example with Anopheles mosquitoes, the ones that carry malaria. Figure 16.01.
Oh, it's a phenomenal example. It really shows the complexity. Researchers looked at three closely related Anopheles species, A. gambiae, A. melas, and A. arabiensis. They sequenced genes from different parts of their chromosome specifically, different chromosome arms, 2L, 2R, 3L, 3R, and the X chromosome.
And what did they find?
They found that the phylogenetic trees built from genes on each of those chromosome arms told a different story about which species were most closely related. Complete conflict.
Wow. So the history is literally different depending on which part of the genome you look at.
Exactly. It was a mess. And this widespread conflict was a result of a long, complicated history of both hybridization and incomplete lineage sorting happening between these mosquito species. Gene flow was just constantly muddying the waters.
So how did they ever figure out the true species relationship?
They made a really critical observation. They noticed that the genes on the X chromosome seemed to show a different pattern, one that looked older, deeper, more consistent.
Why would the X chromosome be different?
Well, in many species, sex chromosomes like the X behave a bit differently during inheritance and recombination. In this case, it seemed the X chromosome was less affected by recent gene flow, recent introgression, compared to the other chromosomes, the autosomes.
Ah, so it held a cleaner historical signal.
It seemed to. The X chromosome gene trees consistently pointed to A. malus and A. arabiensis being sister species, with A. gambiae splitting off a bit earlier. This was taken as the most reliable estimate of the true species phylogeny, the signal that wasn't drowned out by recent hybridization noise.
That's incredible. It shows just how hard it can be to find the species tree when gene trees are all over the place. You have to dig deep, look for the most conserved parts of the genome.
It really does. You often need methods that look at thousands of gene trees and try to find the most common pattern, the consensus history, which we then call the species tree.
Okay, and just briefly, before we move to methods, let's touch on the prokaryotes, bacteria, and archaea. The source says the whole idea of a simple branching tree kind of breaks down for them because of horizontal gene transfer, HGT.
Oh, absolutely. HGT completely changes the game for microbes. This is where organisms transfer genes laterally, directly between individuals, even distantly related ones, not just vertically from parent to offspring.
You're not just inheriting, they're swapping genetic code like trading cards.
Pretty much, and it's incredibly widespread in bacteria and archaea. It means their evolutionary history isn't really a single, clean, branching tree. It's much more like, well, the book uses the analogy of braided hair from figure 16.10, interconnecting strands rather than cleanly diverging branches.
So genes jump between lineages all the time.
All the time. This is hugely important medically, for instance. It's a major way bacteria pick up things like antibiotic resistance genes from completely unrelated species.
Right.
So yeah, while we can still trace the history of core genes, the concept of a single, all-encompassing tree of life for prokaryotes is definitely an oversimplification because of the massive impact of HGT. Their genomes are mosaics of genes with different origins.
Okay, so we've laid out the problems. Parsimony, just counting steps. It's simple, but it gets easily fooled by homoplasy, by reversals, by different rates of evolution. It seems clear why the field needs something more robust. When did they really start shifting towards using explicit statistical models?
Well, parsimony actually worked reasonably well for a long time, especially with morphological data where maybe homoplasy wasn't overwhelming and it's computationally fast. But things really changed when we started getting massive amounts of DNA sequence data in the, say, 80s and 90s.
Right, the genomics revolution.
Exactly. And with that flood of data, the evidence for things like varying evolutionary rates among lineages and the problem of saturation in deep time just became undeniable. It was obvious we needed more sophisticated tools that could handle that complexity. We needed statistical rigor.
So methods that don't just count steps as equal, but actually incorporate the probability of different kinds of evolutionary events happening.
Precisely.
Yeah.
We needed to build models of how characters, especially DNA sequences, are expected to change over time, rather than just assuming the fewest changes is always the best answer.
Okay.
And the two main workhorses of modern statistical phylogenetics are maximum likelihood, ML, and Bayesian inference.
Maximum likelihood. Let's start there. What's the core idea?
Maximum likelihood, or ML, is conceptually really elegant, though computationally intensive. It basically asks this question. Given a specific evolutionary model, like how likely A is to mutate to G, etc., and given the actual DNA sequences we observe in our species today, what is the phylogenetic tree, meaning the branching pattern and the branch lengths and the substitution rates, that maximizes the probability of observing our data?
Whoa, okay. That's a big shift. We're not asking which tree is simplest anymore. We're asking which tree, combined with a model of evolution, makes the DNA sequences we actually see the most probable outcome.
Exactly. It's a fundamentally different question. The source material in box 16a goes into the logic of likelihood. It defines likelihood specifically as the probability of the data, our sequences, given the hypothesis. And the hypothesis isn't just the tree shape. It includes the branch lengths, which represent time or amount of evolution, and the parameters of the substitution model.
Can we walk through their simple example? With just 3 species and estimating 2 branch lengths, T1 and T2.
Sure. Imagine you have sequences from 3 species. You assume a model, say, how fast mutations happen on average. Then, for a particular proposed tree shape and specific values for the branch lengths T1, time to the first split, and T2, time since the split, you calculate the probability of getting the exact sequence differences you observe.
Okay, you get a probability number for that specific tree and those specific branch lengths.
Right. Then you repeat that calculation for a slightly different value of T1 or T2, or maybe even a different tree shape. You do this thousands, maybe millions of times, exploring the space of possible trees and branch lengths. ML algorithms are designed to search for the combination of tree, branch lengths, and model parameters that results in the highest possible probability value, the maximum likelihood estimate.
So in their specific example calculation, they found the maximum likelihood was reached when T1 was 2.7 million years, but T2 was 0. What does a branch length of 0 mean?
A branch length of 0, in that context, would imply that the calculated most likely scenario is that the most recent common ancestor of species 1 and 2 already had the character state seen in both descendants, and effectively no time, or no substitutions, occurred on the branches leading from that ancestor to species 1 and species 2 individually for that specific character being analyzed. It suggests the change likely happened right at or before the speciation event itself. ML finds the parameters that best fit the data, even if those parameters seem a bit weird, like zero-length branches in some cases.
Okay, that makes sense. It's fitting the model to the data, precisely. Now, where does the other big method, Bayesian inference, fit in? How is it different?
Bayesian inference is very closely related to ML. It also relies heavily on calculating likelihoods using evolutionary models. But it has a key philosophical and practical difference. It allows you to formally incorporate prior information into the calculation.
Prior information, you mean like things we already know or strongly suspect?
Yeah.
Like dates from fossils, or maybe known mutation rates from other studies.
Exactly. You can provide the analysis with prior probabilities, information about what you think is reasonable before you even look at the new sequence data. For instance, you could tell it that a split between two groups definitely happened after a certain fossil date.
How does that help?
It can help constrain the analysis, guiding it towards more plausible results, especially when the data itself is noisy or the number of possible trees is astronomically large. Bayesian methods are often better at handling very large, complex data sets where finding the single absolute maximum likelihood tree might be computationally impossible.
And what does Bayesian analysis give you as an output, not just one tree?
Typically, no. Bayesian methods usually explore a whole landscape of pretty good trees and parameters. The output is often a distribution of plausible trees, and the branches are annotated with posterior probabilities, which represent the level of confidence or support for that particular grouping, averaged across all the plausible trees found. It gives you a more nuanced picture of the uncertainty involved.
It's really impressive how sophisticated these methods are. And what's reassuring, I guess, is that despite all this complexity, the different methods, the potential conflicts in data, the overall picture of life's history often seems to hold up, right? Especially when you compare trees built from morphology, like bones, with trees built from molecules like DNA.
Yes, that general congruence between different data types and different methods is incredibly important. It's why scientists have high confidence in the broad outlines of the tree of life. When DNA-based phylogeny is analyzed with ML or Bayesian methods, largely confirm or maybe just refine the relationships that were first proposed based on careful anatomical study, that's powerful corroboration.
And this convergence of evidence was crucial for finally settling some long-standing debates, like the big one about birds and dinosaurs. That's figure 16.110.
Absolutely. The bird-dinosaur link is a fantastic example. The morphological evidence, the synapomorphies based on bones, was actually there for a long time. Things like, well, the cubus bone pointing backwards, hollow long bones, fused clavicles forming a wishbone or furcula,
Right, the wishbone.
and even the very specific structure of the hand having only 3 digits. The source points out how the hands of the early bird Archaeopteryx and the non-avian dinosaur Deinonychus are remarkably similar, almost identical.
But still, there was resistance, right? Some paleontologists were hesitant to fully accept birds were dinosaurs.
There was. For a long time, birds were kept in their own class, aves, separate from reptilia. Partly because the evolution of flight and feathers seemed like such a massive, unique jump. It made them feel fundamentally different.
So what sealed the deal?
It was really the flood of molecular data, analyzed with these robust statistical methods like ML and Bayesian inference. The DNA evidence overwhelmingly confirmed the signal from the bones. It placed birds firmly inside the theropod dinosaurs, a specific group that includes things like Velociraptor and T-Rex.
So the anatomy and the DNA told the same story.
Exactly. The combination of the detailed anatomical comparisons with the statistical power of the genetic analysis just became undeniable. It was a major intellectual triumph for phylogenetic systematics, resolving decades of debate and confirming that birds are, in fact, living dinosaurs.
Okay, so now we have these robust trees, built with sophisticated methods, often supported by multiple lines of evidence. We know who is related to whom with pretty high confidence. The next big step is figuring out when these splits happened. This brings us to the molecular clock.
Right. The molecular clock is a really powerful concept. The basic idea is pretty simple. If mutations accumulate in a gene or a DNA sequence at a relatively steady average rate over long periods of time,
Like a clock ticking.
exactly, then the amount of genetic difference, the sequence divergence between two species, should be proportional to the time since they last shared a common ancestor. More differences equals more time has passed.
It sounds almost too good to be true. Like using radioactive decay, but for genomes.
Conceptually, yes, it's a brilliant idea. But, and this is a huge but, the core assumption of a perfectly steady, constant rate is often violated in reality.
So the clock isn't always reliable.
Rates of sequence evolution can actually differ quite dramatically across different groups of organisms. We touched on this earlier with differential rates. For example, molecular evolution tends to be slower in many primates, including us, and also in things like trees and shrubs, compared to how fast it runs in rodents or herbaceous plants.
Wait, if the clock ticks at different speeds in different lineages, slower here, faster there, how can we possibly use it to tell time accurately? Doesn't that kind of break the whole concept?
It certainly complicates it. It means we can't just assume a single universal clock rate for all of life. We have to account for that rate variation. Modern methods try to model this variation, allowing rates to change across the tree, but we also need to understand why rates might vary.
Is there a leading hypothesis for that?
One major explanation relates to generation time. The idea is that species with shorter generation times, like mice, which reproduce quickly, go through more rounds of DNA replication in their germ cells, sperm and eggs, per unit of absolute time, say per year.
More replication cycles means more chances for mutations to occur during copying.
That's the thinking. So over the same number of years, a lineage with rapid generation turnover might accumulate more mutations than a lineage with very long generation times, like elephants or humans.
So it's the number of generations, not just years, that might be key.
It seems to play a big role, but it's probably not the whole story. Other factors like metabolic rate or the efficiency of DNA repair mechanisms could also influence the background mutation rate. We know, for instance, that primates generally have slower molecular clocks than many other mammals, and that's likely due to a combination of longer generation times and maybe other physiological factors.
Okay, so given all this variation, just assuming a constant rate seems really risky. We must need some way to anchor these estimates, right? Some fixed points in time.
Absolutely essential. And that's where calibrating the clock with fossils comes in. Fossils are crucial.
How do they help?
Fossils provide minimum age estimates for specific divergence points on the tree. If you find the oldest known, unambiguous fossil that clearly belongs to a particular group, say the oldest fossil bird,
Right.
then you know the split between birds and their closest relatives, other dinosaurs, must have happened at least that long ago. It couldn't have happened more recently. These fossil dates act as calibration points, like pins on the timeline.
So you use these known minimum ages to figure out the actual rate of molecular evolution along specific branches.
Yes, or more often now, you incorporate these fossil dates as constraints within the sophisticated statistical dating methods, often the Bayesian ones we mentioned. These methods can then estimate the divergence times while allowing the evolutionary rate to vary across the tree, but keeping everything consistent with the fossil evidence.
Let's look at a really important example, dating the split between humans and chimpanzees. That's figure 16.1 sin. How was that estimate derived?
That's a great case study. One major study mentioned in the text used a very large amount of DNA sequence data, almost 60,000 base pairs. Critically, they calibrated their molecular clock using 4 well-established fossil dates from different points in the primate family tree.
Okay. Lots of data, multiple fossil anchors.
Right. And their analysis, using methods that accounted for rate variation, estimated the human-chimpanzee divergence time at approximately 6.6 million years ago, Maya. They gave a pretty tight statistical range, too, something like 6.0 to 7.0 Maya.
And has that date held up? Have other studies confirmed it?
Remarkably well, yes. The confidence has actually increased because different independent approaches have arrived at very similar numbers. For instance, another more recent analysis used whole genome sequences, even more data. But instead of relying primarily on fossils, they estimated the split time based on direct measurements of the human mutation rate per generation, and then factored in generation times.
A completely different approach to calibration.
Totally different. And that study estimated the human-chimp split at around 6 Maya. The fact that these complex independent methods, one anchored mostly by fossils, the other by mutation rates, give such similar answers, provides really strong confidence that the split happened somewhere in that 6 to 7 million year ago ballpark. They also estimated the split between the human chimp lineage and gorillas happened a bit earlier, around 8.6 to 10 Maya, again with good agreement between studies.
That's really powerful when independent lines of evidence converge like that. It's also fascinating that these phylogenetic dating methods aren't just for biology. The source mentions they've been borrowed by linguists and anthropologists.
Yeah, it's pretty cool. Charles Darwin himself actually noted the analogy way back. He saw how languages seem to diverge from common ancestral languages, much like species diverge from common ancestors.
So linguists use tree-building methods now.
Widely. They use phylogenetic algorithms to reconstruct language family trees, tracing how different languages evolved, split off from each other, and spread geographically over time.
The book gives the example of the Austronesian languages to show how this can even model cultural evolution. That's figure 16.19.
Right, the Austronesian language family is enormous. It originated in Taiwan probably over 5,000 years ago and then spread incredibly widely across the Pacific and Indian Oceans. Think languages in Madagascar, the Philippines, New Zealand, Hawaii.
Well, no, huge distribution.
Huge. So researchers built a phylogeny based on the relationships between the languages of 84 different Austronesian societies. But then they went a step further. They mapped cultural traits onto this language tree, specifically looking at how political organization, things like chiefdoms, states, changed over time within these cultures.
So using the language tree as a proxy for the cultural history.
Exactly. Assuming, reasonably, that language and culture are often inherited together. And they used phylogenetic comparative methods, similar to what biologists use, to test hypotheses about how political complexity evolves.
And what did they find out about how power structures change?
The results were really interesting and felt, well, realistic. They found that political organization tends to increase in complexity only through small, incremental steps, building up institutions, hierarchies, stability that seems to be slow, gradual work, evolutionarily speaking.
Okay, makes sense.
But, and this was the key contrast, they found that political organization can decrease in complexity either by small steps or by large, rapid steps. Collapse or simplification can happen much more quickly and dramatically than building up.
Huh. Easier to fall apart than to build up.
Seems that way, at least based on this analysis. It really demonstrates how these phylogenetic tools, originally developed for biology, can provide powerful insights into patterns of cultural inheritance and evolution, too.
Okay, moving on to some applications that really push the boundaries. Using phylogenies to actually look back in time and figure out what ancient, extinct organisms were like. This is reconstructing ancestral traits. Feels a bit like biological time travel.
It kind of does. Ancestral state reconstruction is definitely one of the most intellectually exciting uses of a phylogeny. You use the information from the living species, the tips of the tree and the tree structure itself, to statistically infer the characteristics of their long-dead common ancestors at the internal nodes of the tree.
So you can predict ancestral features.
Yes. And for molecular biologists, this gets really cool. You can infer the likely DNA or protein sequence of an ancestral gene. Then, and this is the amazing part, you can actually synthesize that inferred ancestral sequence in the lab, put the gene into cells or bacteria, produce the ancient protein, and study its properties. You can literally resurrect ancient molecules.
Wow. Okay, the case study on fish vision, the opsin proteins, sounds like a perfect example of this whole process. That's figure 16.20.
Oh, it's a fantastic study, truly elegant. Researchers were interested in rhodopsins. These are the main proteins responsible for vision in dim light. They were studying fish that had adapted to live in really deep, dark ocean environments.
Okay, deep sea fish.
Right. Now, in the deep sea, the little light that penetrates is mostly blue. So for fish to see well down there, their rhodopsin needs to be most sensitive to blue light, meaning its maximal absorption wavelength, called lambda max, needs to shift towards shorter, bluer wavelengths compared to shallow water relatives.
Makes sense. Tune the eye to the available light. So how did they use the phylogeny?
First, they built a phylogeny of these fish, including both deep-sea and shallow water species. Then, using the rhodopsin gene sequences from the living fish, they statistically reconstructed the likely sequence of the rhodopsin protein in the common ancestor before the lineages moved into the deep sea.
They figured out the ancestral protein sequence.
Yes. Then they synthesized that ancestral gene, expressed the protein, and measured its properties. They found its lambda max was 501 nanometers. That's sort of greenish-blue light, typical for surface or mid-water vision.
Okay, that's the starting point. But the modern deep-sea fish, like the conch eel mentioned, they need to see even deeper blue light. Really?
Exactly. The modern conger eel's rhodopsin has a lambda max shifted down to 486 millimeters, much bluer. So the researchers then looked at the specific amino acid changes that happened along the evolutionary branch leading from that 501 millimeter ancestor to the modern conger eel. They wanted to know which mutations caused that 15 millimeter blue shift.
Did they find a single magic mutation?
That's what they tested. They made proteins with individual mutations, but here's the critical finding. No single mutation could account for the full shift. It turned out that 3 specific amino acid substitutions, which happened along that lineage, were required together simultaneously to achieve the full shift down to 486 nanometer function.
Ah, so the aha moment wasn't just finding the mutations, but realizing they had to work as a team.
Precisely. It revealed the importance of epistasis, which is when the effect of one mutation depends on the presence of other mutations. Synergism. A single substitution might have done very little on its own, maybe even been slightly bad. But when all 3 were present together, they interacted to produce the significant adaptation, the enhanced blue vision.
That's a really profound evolutionary insight. The adaptation isn't always just adding up small effects, but can involve complex interactions.
Absolutely, and that conclusion that the full adaptation required this specific combination of mutations working together would be incredibly difficult, maybe impossible to figure out without using the phylogeny to reconstruct the ancestral state and trace the specific historical path of the mutations. It shows the tree isn't just descriptive, it's an experimental tool for understanding how evolution works.
That really drives home the power. It's not just history, it's mechanism.
We see similar power in other areas too, like tracking diseases. Figure 16.21 mentions using phylogenies to understand the spread of the Ebola virus during an outbreak. By quickly sequencing virus samples from different patients and building a phylogeny, health officials can figure out how the virus is moving, where it came from, maybe how many separate times it jumped from animals to humans. That information is vital for guiding containment efforts.
Right. Using the tree for public health forensics. Another key application mentioned is using the phylogeny almost as a statistical control, the comparative method for studying adaptation. What's the goal there?
The comparative method is all about testing hypotheses about why traits evolved. It looks for correlations between a particular trait and some environmental factor or aspect of the organism's lifestyle. But crucially, it only considers a correlation to be evidence for adaptation if that correlation has arisen independently multiple times on the phylogeny.
So we're looking for evolution repeating the same pattern, nature's independent experiments.
Exactly. We want to see if, whenever a certain environmental pressure appears, a particular trait tends to evolve in response across different unrelated groups.
The example given is about testes size in relation to mating systems. That sounds interesting.
It's a classic example. The hypothesis is pretty straightforward. In species where females mate with multiple males, polygamous or promiscuous systems, there's intense competition between the sperm from different males to fertilize the eggs.
Sperm competition, right.
So the hypothesis predicts that natural selection should favor males who produce more sperm in these competitive situations. And one way to produce more sperm is to have larger tests relative to your overall body size.
Okay, makes biological sense.
Yeah.
And do we see that correlation?
Yes. When researchers compiled data across many different animal groups, mammals, birds, fish, they consistently find that species with polygamous mating systems tend to have significantly larger tests for their body size compared to closely related species that are monogamous, where females mate with only one male.
Okay, so the correlation is there. But why do we absolutely need the phylogeny? If we just make a big list of species, their mating system, and their testes size, won't the correlation just pop out?
Ah, but that's where you can get fooled. You absolutely need the phylogeny to act as a statistical control, specifically to avoid being misled by shared ancestry or phylogenetic inertia.
Meaning?
Imagine you're comparing 2 species of monkeys that are very close relatives. And let's say both happen to be polygamous and both happen to have large tests. If you just put them in your spreadsheet as 2 separate data points supporting the correlation, you're statistically inflating your evidence.
Because they probably just inherited both traits from their recent common ancestor. It's not 2 independent evolutionary events.
Exactly. It's really only one data point reflecting that ancestral condition. The phylogeny allows us to identify true independent evolutionary contrasts. We look for points on the tree where a lineage changed its mating system, say from monogamous to polygamous, and then we ask, did its relative testes also change in the predicted direction at or after that point? That's an independent evolutionary event.
I see. You're comparing sister groups that differ in the trait and the proposed selective pressure.
Precisely. The source gives that great warning example, comparing birds and mammals. Birds fly and lay eggs. Most mammals don't fly and have live birth. That's a perfect correlation. Flying goes with egg laying. Not flying goes with live birth.
But that correlation is totally meaningless, right?
Completely meaningless in an adaptive sense. It's entirely due to deep ancient history. The common ancestor of birds flew and laid eggs, or at least was on that trajectory, and the common ancestor of placental and marsupial mammals was terrestrial and had live birth, or was on that path. There's no functional or adaptive link between flying and laying eggs that caused them to evolve together repeatedly. It's just evolutionary baggage inherited by entire huge clades. Phylogenies are essential to statistically control for this shared history and isolate true instances of correlated adaptive evolution.
Okay, wrapping up now. Let's look at how phylogenies have fundamentally reshaped how we classify life, the whole field of systematics. What's the absolute core principle driving modern biological classification?
It's actually very simple and very strict. A modern classification must be based directly on phylogeny. The ultimate goal is that every officially named group, every taxon, whether it's a species, genus, family, order, whatever, must be monophyletic.
Monophyletic, we heard that earlier. That means it has to be a complete clade, right?
Exactly. A monophyletic group, a clade, includes a common ancestor and all of its descendants. No exceptions. If a named group isn't monophyletic, it's considered an invalid artificial classification because it doesn't represent a single, complete branch of the evolutionary tree. It doesn't reflect a natural unit of history.
So let's nail down the definitions of the bad groups again, the ones we need to avoid. We need monophyletic. What are the others?
Right. The two main types of non-monophyletic groups to avoid are paraphyletic and polyphyletic.
Paraphyletic first.
A paraphyletic group includes a common ancestor, but, and this is the key, it excludes some of the descendants that belong to that same branch.
So it's like drawing a boundary partway up a branch, leaving off some of the twigs at the end, maybe the ones that look really different.
That's a good way to think about it. You're recognizing an ancestor and most of its descendants, but deliberately leaving out one or more subgroups that actually evolve from within that main group.
Okay. And the other bad one, polyphyletic.
Polyphyletic is generally considered the worst offense against evolutionary principles. A polyphyletic group includes species that are descended from more than one common ancestor. The group is based on some similarity, usually a convergent trait, homoplasy, that makes them look alike, but they don't actually share a unique, recent common history. You've mistakenly grouped unrelated lineages together.
Got it. Okay, the classic example used to illustrate a paraphyletic group is the traditional concept of reptiles versus birds. That's figure 16.24.
Yes, it's the textbook case. Historically, the class Reptilia was defined to include turtles, snakes, lizards, crocodiles, but it specifically excluded birds, which were put in their own separate class evs.
Even though we now know birds evolved from dinosaurs, which are reptiles.
Exactly. The phylogeny makes it absolutely clear. Birds evolved from within the dinosaur lineage, specifically the theropods. Dinosaurs, in turn, share a common ancestor with crocodiles more recently than either shares with lizards or snakes. So birds are deeply nested within the group that was traditionally called Reptilia.
So if you go back to the common ancestor of, say, a crocodile and a lizard, you find that birds are also descendants of an ancestor within that group.
Precisely. To make the group containing crocodiles and lizards and snakes monophyletic, you must also include the birds because they are descendants of that same ancestral stock. By leaving birds out, the traditional Reptilia is paraphyletic. It's an artificial group defined by shared ancestral vertebrate traits like scales, cold-bloodedness, and by excluding the highly modified, feathered descendants.
So how does modern classification handle this?
Modern classification demands monophyletic groups. So biologists now use terms like Sauropsida, which includes all traditional reptiles plus birds, or Archosauria, a smaller monophyletic group including crocodiles, dinosaurs, and birds, to reflect the actual evolutionary relationships. The old paraphyletic Reptilia is generally avoided in formal phylogenetic classification.
Okay, that clarifies paraphyletic. What about an example of a polyphyletic group that got corrected?
A great example is the historical bird order Falconiformes. For a long time, this order was used to group together falcons, hawks, and eagles.
Makes sense. They all look like birds of prey. Hooks, beaks, sharp talons.
Exactly. The grouping was based entirely on those shared adaptations for predation. Powerful examples of convergent evolution driven by a similar lifestyle. They look superficially very similar.
But what did the DNA evidence reveal when people applied those modern ML and Bayesian methods?
It revealed a huge surprise, completely overturning the traditional view. The molecular data showed that the similarities were purely superficial, just homoplasy. Falcons are actually much more closely related to parrots and songbirds, passerines, than they are to hawks and eagles.
Wow, really? Falcons and parrots.
Yes, and the hawks and eagles, they form their own separate, distinct monophyletic clade, which is now called
So the
accipitriforms.
old falconiforms, which lumped falcons together with hawks and eagles, was bringing together members of two very distant, independently evolved lineages just because they looked alike.
Precisely. It was a classic polyphyletic group based entirely on convergent traits, not shared ancestry. Modern bird classification has completely revised this now. The order Falconiforms is restricted to just the falcons and their immediate relatives, a monophyletic group, and the hawks and eagles are in their own order, accipitriforms.
It really underscores the importance of relying on the phylogeny.
Absolutely. This strict insistence on recognizing only monophyletic groups in classification ensures that our naming system actually reflects the true evolutionary history of life, not just human convenience or potentially misleading appearances. Hashtag tag outro. You know, what's really striking when you step back is that a phylogeny isn't just a diagram. It's truly an intellectual tool. It's a dynamic, testable hypothesis about history. It's not some dusty old family tree locked away in a museum.
Yeah, it's active.
It's the framework that lets us run these incredibly powerful analyses, like we saw dating the human-chimp split, or literally recreating the vision of ancient fish to understand how their proteins evolved, proving things like evolutionary epistasis. It's amazing.
Okay, so let's distill this down for the final takeaway. Today, we really dove into phylogenies. We learned that are these crucial hypotheses about evolutionary history. And they're not just about who's related to whom, but they allow us to date ancient events, to reconstruct what ancestors were like, even their proteins. And critically, they explain why modern biological classification absolutely insists on recognizing only monophyletic groups, only complete branches of the tree. Thinking critically about how traits relate to ancestry, it's just fundamental to all of biology.
And maybe the bigger picture connection is this. When you look at any living thing now, any animal, any plant, you know that every single feature, it has its anatomy, its molecules, its behavior. You can analyze it from two perspectives, right? There's the mechanism. How does it work right now? And then there's the history. How does it get that way through evolution? Phylogeny is the key to unlocking that historical perspective. But as we saw, that history isn't always a simple, neat story.
Right. And that leads to what I think is a really fascinating concept to ponder from today's discussion. Thinking about things like incomplete lineage sorting, and especially horizontal gene transfer. It really shows that the history of life isn't always this perfect bifurcating tree, is it?
Definitely not, especially for microbes.
That idea of a braided hair phylogeny for prokaryotes, and even the conflicting histories we saw mapped onto different chromosome arms in those Anopheles mosquitoes, it raises a really profound question, doesn't it? If individual genes can have genuinely different histories, how often is the species tree that we reconstruct just a statistical average, maybe obscuring a whole bunch of conflicting underlying truths? It makes you wonder if maybe the very concept of a single universal tree of life needs rethinking, perhaps moving towards a more complex web or network, especially when we consider deep time in microbial evolution.
That is indeed a provocative question. How much of the history is tree-like versus network-like? It's an active area of research and debate.
Something for us all to think about. Okay, next time, we're going really big picture. We're going to take a grand tour through the entire history of life on Earth. We'll cover the key geological context, the stage upon which life evolved, and hit the major evolutionary transitions across the vast expanse of geological time.
Yeah, get ready for a journey through the eons. We'll be covering about 4 billion years from the Archean origins right up through the major eras to the Cenozoic. It should be quite the ride.
Looking forward to it. Thanks for listening to Bookfulness. Join us for the next deep dive.